This is a presentation that I gave at the Joint Bioenergy Institute. It's related to a competition on Kaggle called the Open Problems Multimodal Single Cell Integration Competition. And the goal was how to predict DNA, RNA, and protein measurements and how they co-vary in single cells. So this is brand new data. And we're going to be having a look at it, and especially we're going to be focusing on the power of PCA. So the table of contents, we have an introduction, challenges, data description, exploratory data analysis, model building, feature selection with a spotlight on the time series nature of the data, and some conclusions. One of the questions we'll be asking ourselves is, can we predict differentiation of stem cells? How much can we do with single gene data? And how many proteins have we quote-unquote cracked? So let's get started. So the goal of the competition was to predict how DNA, RNA, and protein measurements co-vary in single cells as bone marrow stem cells develop into more mature blood cells. And so we had over 300,000 data points here on CD34 plus hematopoietic stem and progenitor cells from 4 donors at 5 time points that were generated by celerity. And so we have 3 layers of information. This is showing the first 2 layers, which is the first one is accessibility, gene accessibility, and the second one is gene expression. And here the task is to predict gene expression from gene accessibility. And the metric is how well your predictions correlate with the true expression levels. All right, so some history is that in the past decade, the advent of single-cell genomics has enabled the measurement of DNA, RNA, and proteins in single cells, and have allowed the study of biology at an unprecedented scale and resolution. And among the outcomes have been detailed maps of early human embryonic development, discovery of new disease-associated cell types, and cell-targeted therapeutic interventions. And so we can now even measure multiple genomic modalities in the same cell. So as you can see, we can measure always 2. So we can measure accessibility with expression and expression with protein levels. So the task is to, given A, predict B. There are 2 sets. The multiome data set, the multiome kit, measures the accessibility and the gene expression, whereas the CITESeq kit measures gene expression and surface protein levels. And so we have the central dogma of molecular biology, that is, given DNA, predict RNA, and then given RNA, predict protein levels. And so that's going to be our task here. And so this is an overview of the experimental procedure. On day 1, all the cells were thawed and plated. And then days 2, 3, 4, 7, and 10 each had observations taken. Day 10 had multiome data as well, whereas for the CITESeq data, that data was unfortunately not available, which is a little bit of a problem because you don't have, you only have 1 3-day jump in the CITESeq data. So you cannot really predict that jump very well, since before you only had 1-day jumps. All right, so here's the train and the test splits for this competition. They're very interesting, completely non-random selections of the test set. So the test set was a new donor. First of all, what you can see during the competition time while you're working on the competition, and the final scoring of the competition was on a new day. So the Pearson correlation coefficient was used to rank the submissions. For each observation, the correlation between the ground truth gene expressions and the predicted gene expressions is computed, and the protein levels similarly. So the idea is that you have tens of thousands of gene expressions for each cell in the target, right? And so um, how do you compute one number figure that tells you how well you did? And so the correlation is a way to do that. So this correlation is always a number between -1 and +1. +1 means you're correlating perfectly, meaning you basically always go up when the ground truth goes up, and you go down when the ground truth goes down by the same fraction, by the same percentage. It is not sensitive to the raw levels, so you could shift all your predictions by constant or inflate them by a factor of 2, and it would not change the correlation. So there are, in total, I believe, over 1,200 teams competing, and these are the prizes. So the public leaderboard was extremely close, given that many grandmasters made their solutions available. And so at the end of the day, the private leaderboard was not so close. So the private leaderboard had the top 3, which were each different by 0.1. But on the public leaderboard, people go between 0.001% is the kind of maybe a figure of merit for the difference. So challenges, there are a number of challenges. First of all, here we have time series data. So whereas the current pipelines treat cells as static snapshots, here there's an underlying dynamical biological process, for example, differentiation, right, of stem cells. And so that's an open challenge. And so one of the things that we can do here, they did give us some metadata. So the cells do change over time. And so as you can see, on day 7, there's a different mix of cells present as there was on day 2. And the same is also true for the multiome data. So you can see here that one of the goals of this competition was to predict changes in genetic dynamics over time. And so again, here you can see, using UMAP, that different regions of genes were expressed during the different days. So blue here is earlier and red is later, and you can see that there are different things happening on the left side for the gene expressions and on the right side for the protein levels. The next thing is that there's gene regulation taking place, which makes it difficult for later days. Here we have feedback, which is that proteins that are produced may circularly bind back to DNA and inhibit the process. So typically this is the central dogma of molecular biology, but here you have a feedback that the proteins can interact with the DNA directly. And so this is actually something that we cannot accurately measure, because we don't have protein and DNA data, right? We have DNA and RNA data, and we have RNA and protein data, but we don't have protein and DNA data. So this is mostly a noise. This becomes noise, and as you will see, as we go further and further into day 7 and day 10 even, there's more and more noise. And this may be one of the reasons. All right, so there are batch effects. I'll go into the slide in greater detail at a later point here, but the green bars are for day 3. So the red ones are day 2, the blue are day 4, and the yellows are day 7. And what we're plotting on the left is the sums of all the gene availabilities for one cell. So you take all the genes, right, 200,000 plus genes, you sum up all the availabilities, and you can see that for some reason on day 3, things were happening more. And the question is this something that's relevant? Maybe, right? It seems it should be maybe relevant. But is it, random, a random technical effect? basically you always have to worry. So, you know, at the end of the day, you will see that these type of uh, shifting effects where the totals are all just randomly bigger don't actually play much of a role because the correlation is insensitive to um, ballooning. Like you can predict twice for everything and then you'll still get it completely correct. So that's maybe one of the downsides for using correlation, but at the same time, using something like mean square error, you wouldn't have this effect, but it would also be, it would just be a different metric that you're computing at that point. All right, so for the size of the data, we have some big data sizes here. So we have for the multiome input, train data 100 gigabytes. So really big data, but we do have the ability to use sparse arrays. So sparse arrays are very useful. They just double the number of um, floats you have to store for each point, but you don't have to store any of the zero values, right? So very useful, especially for the multi-ohm data. But another one of the reasons um, that working with this data is difficult is, you can't really have 200,000 plus genes as input when you only have 20,000 outputs, right? That's overdetermined. You can easily match that by completely overfitting. And so one of the things that you can do is you can use PCA, and in our case, singular value decomposition, because that works with sparse arrays. But also luckily we are able to have high RAMs and high storage via Saturn Cloud that was made available to us for free as part of this competition. So we have very fast computers, very fast GPUs with lots of RAM and lots of storage, which is absolutely essential for doing this type of competition. All right, so the dataset description, let's have a look at the data collection here. We have 4 donors. They were incubated, media was changed, hopefully nothing went bad. Here's the names of the surveys that were done, the CITESeq for the gene expression and protein expression, and then the ATAC-seq for the gene availability and gene expression. All right, so we have the first file was given to us was the metadata file. It has the day, the donor, and the technology, first of all. So technology is one of 2, CITESeq or Multiome. And then it also gives us the cell type, which is another description here that they tell us it's kind of incomplete or it's imprecise. It's basically a way to label cells and tell you what they are. Turns out I don't think anybody used this. But it is interesting because, as I showed you before, it really shows you that things are changing over time. So it's 2 competitions in one. You can one how to encode these features. I don't think anybody did. All right, so for the multiome data, here's, for example, how you would write this, or how you would access this data. You have the column names, and so for the input data, which is on the top, there's 228,942 columns, and these are the names of the columns. So each of them has the name of the chromosome, and then it has a start and an end point on the chromosome, which is the identifier for this particular gene. There are over 200,000 of them, and you're predicting RNA expression data. And so those are given as these codified blocks where there's 4 letters and then a bunch of numbers, and so that's the RNA target data. And then for the CITESeq, similarly, you're starting with that. So you can see it's essentially the same, but it gives you some more information. And the reason it does that is because what's after the underscore can be relevant. Sometimes they put the name of a protein, a particular protein, after the underscore. And so that means that that RNA has previously already been identified to have particular relevance for this particular protein. And so that's one of the reasons why they put that underscore. And then, so I already told you that there are some cell type annotations here. So there are in total 7 cell type annotations. They change a lot over time. And so let's start with the exploratory data analysis. So this is by Ambrose M. I took all of his slides or all of his figures, and then I made my own comments. So here you can see some distributions. It's pretty evenly distributed overall. Nothing really too special. Similarly, you can break it down how many cells there are per day, and donor, again, evenly distributed. On day 4, one of the donors did not pass quality control, so the bar is missing there. Non-zero entries, you can see the non-zero entries of the Multiome data, where we just look at all the data of all the cells. Is you know, has outliers. There's some numbers at 8, but it's a pretty regular distribution. There's nothing too crazy in terms of discreteness or anything. Similarly with CITESeq, one last thing is that the Multiome data, apparently there's probably some experimental sensitivity, so the data starts at 3 rather than 0. So basically everything below 3 is binned into the 0 bin. Probably the experiment just can't recognize it, and so you have some artifacts of the experiment not being maybe completely precise. For CITESeq, it seems like it's more of a continuous curve that goes all the way to 0. And so indeed you will see that the CITESeq data for us is more predictive than the Multiome data. So there's here a spy plot where you can see that the non-zero values are concentrated. So there's these bars. Some of the gene expressions have lots of non-zero, and some have very few non-zero entries. And so, for example, here are a few of the input for the CITESeq data. You can see that there are all kinds of distributions for the different gene expressions. So there you could have continuous, you could have discrete, kind of like a bar plot, multimodal, 2 peaks or just 1 peak. All are positive. And so it turns out one of the things that I tried is changing the 0 and replacing it with a mean, thinking that, oh, if it doesn't know, it's just going to put the 0, and so it's better to use the mean, but that's actually not true. So 0 is actually better to be left at 0. And so you can see here over the days and the donors, there's some change here. And so over time, it looks like the plots are getting more filled out or a little more broadened. And then the different donors seem to also have very different plots. And so here you can see that on day 2 there was a mistake made by Celerity, that the 3rd and the 4th donor have exactly the same cells. So they made a mistake there. And then looking at the protein concentrations, again, we see a bunch of different ones. We see normal, bimodal, discrete, outliers all over the place. So it's just a complete mess. Right? Predicting these protein concentrations is going to be difficult. And again, doing the same thing, things change over time and with the different donors as well. All right, so here, some people on Kaggle notice that after the underscore, you have the names of proteins in 151 cases. For example, Pucho only used 84 of those. But in general, you could basically take those aside and give them a special place. If you do special transformations to all the other things, maybe don't do it to these, and then just use these as inputs directly, so that it kind of preserves that significance that had previously been established. And so again, you have these batch effects. And so the question is always, is this an artifact of the technical procedure, or is this a real phenomenon? And so one of the things you can do is you can normalize the data per batch, and then feed them in together, um, and see if that improves it. Turns out I don't think necessarily makes anything better. And you can also just build a new model for each day. And then you don't need to worry about it, right? So the greens have a different model than the blues and the reds. You can do that too. And so here again, you can see in the Multiome data, there's something strange about the yellows, which is a problem because those are the ones we need to predict. So 2, 3, 4, 7 is the, well, actually, 7 is not the one we need to predict. 10 is the one we need to predict, but 7 is on the way towards 10. So you can kind of think things are going to be more crazy even with 10 than 7. So, but in general, the overall normalization is not factored into this. The Pearson correlation does not care about the overall normalization of your prediction. So we have one female, 3 males. That can also be one-hot encoded, and in fact, I don't think anybody did. Model building. So this is again a solution by Ambrose M. Dimensionality reduction. We're going to do SVD, which turns out to be absolutely marvelous. You can't do better than that, even if you include all the raw features. And we're going to have a multi-layer neural network. We're going to have a loss function, which is the Pearson correlation coefficient directly. So that's nice because that's directly what we want to predict for the competition. And so that's something that's actually a differentiable function. Coded by Lucas Morin, hyperparameter tuning with Keras tuner, and then some cross-validation with group k-fold. So here is a slide that shows that you don't need to use a lot of PCA components. So if you just use the Y data only, you don't use the X data, you can already get pretty good correlation with the target by just always predicting the mean. So you predict the average protein concentration for every cell, and then for every kind of protein, and then that'll give you already 72% correlation. And it turns out using 4 PCAs, you can almost get all the way there. So you don't really need a lot of PCAs. And in fact, after 64 PCAs, you don't gain anything. The only way you gain at the end is if you use these special features, the 151 features that were previously identified, and you slap them onto the PCAs. You get some improvement. So that kind of shows that the cell, during this healthy operation that we're dealing with here, these healthy donors, it does not really use more than 64 knobs to regulate itself. And those are reflected by the PCA. And so here, basically, there are some columns you need to take out that are always constant, some columns that have special importance, and the rest gets SVD'd to just 64 components. So all this, tens or hundreds of thousand dimensional data gets pushed together to just 200 or so. And we're going to use some dropout. We're going to use regularization, and we're going to use 3 hidden layers to 56, 512, and 512 for the size. So similar to the size of the input. There's going to be cross-validation. So using group k-fold, so you predict on 2 days, or you train on 2 days, you predict on the 3rd day. So and then you cycle around, making sure it's robust to predicting on other days. And you get a pretty good correlation of 89%. And so the question is, what's going to be on day 7? So here, as you can see, the performance on day 4 is a little bit lower. So it's a little worrying that day 7 is going to not be quite as good. And let's do the same for the multiome. So here we can also do the same for the multiome. Turns out we only need 40 PCA components for the multiome input to predict the output. And so what we do is we predict the PCA of the output, and then we inverse PCA that. And you can see some worrying trends. Day 7 is not good. So day 7 is something we did not have for the CITESeq, but it's not good for the multiome. And so let's look at feature selection. So the data structure is that there's DNA, RNA, and proteins going from huge 200,000 plus to just a couple hundred for proteins. And then, however, the model goes the other way around. So there are 40 knobs at the DNA level going to 64 knobs, and then we shuffle in the 151 special columns, and then going to 140 proteins. So the model is completely the other way around. It goes from small to large, whereas the gene data goes from large to small. And so one of the questions you might ask is, well, what could we do better with more data? And so here, if you use less data, just like 10 points or 100 points or 1,000 points, you can see that indeed you start to improve. And then when you use multiple donors and days, and you start to predict different donors or different days, then you start to flatline, which is good. It means you're more generalizable, right? You can now be sure this model is no worse, and it also can do different donors and different days. But at the same time, if you had more data for each donor, this curve does not look like it has plateaued in the white region. It could still go up more. And so the idea is more data would still help, but it's a log plot, so 10 times as much. So you're looking at a terabyte of data. And for the multiome data, here we do 2 different things. One is we can just predict the PCA, or we can predict the after the inverse PCA. So you can compute the correlation in either case, and it turns out to give completely different numbers. So the PCA, if you're using the RNA only as an intermediate step to go from DNA directly to protein, because the PCA is all you need as input for the protein, right? But if you're interested in RNA in its own right, you're going to inverse PCA that, and then you're going to compute the correlation after. And so turns out the PCA gives you almost 99% accuracy, but most of that, like 96% of that was already available through just using the means. So you don't need any gene, any X data at all to predict that. And then using genes, you can get another 3%. And with the correlation, the numbers are closer to 64% for using no genes, so just the averages, and then it goes up to 68, so you have a 4% gain from actually using some input data, some X data. And so as you can see here, the multiple donors are pretty much flatlined. You can predict different donors pretty well. If you're predicting different days, both the PCA and the correlation prediction goes down significantly. And so let's have a look at that. That seems kind of strange. And so one of the questions I was asking myself, okay, I cannot predict a different day very well. And the problem we already identified is with day 7. So here you can see I'm always using 3 days to predict a 4th day, so day 2, 3, and 4 to predict day 7, and so on, and I do terribly, as we already thought it would happen. And then I compare that to using same day, same donor data to predict. I was asking myself, well, maybe the cells have just gone bad on day 7, and I cannot even predict other cells based on cells from the same day and donor. And that turns out to be partly true. However, gone bad here just means I can't predict it. It possibly means that there some feedback from the proteins that I don't know about because I don't have the protein data. And so that could be that this gone bad is just an artifact of that, and that is introducing all this random noise. But it so turns out that it does not actually affect the top 64 PCA components. So I can perfectly well describe the top 64 PCA components on day 7 using same day data. And so that's actually kind of interesting, right? So it means that the cells really have changed on day 7. You can no longer predict the PCA, which is what gives you then the protein data, on using days 2, 3, and 4, but you can do it using day 7. And so here, you kind of wonder, well, what are these 64 PCAs? Have they changed a lot? So are the day 7 PCAs just wildly different from the other days? And so here we show the overlap between different PCAs. So for example, here on this second plot in the middle, you can see using data from day equals 4, predicting data from day equals 7. So the first PCA component, which is the little white dot up in the left corner, shows that the first PCA component is the same between day 4 and day 7. Whereas the second PCA component on day 7 is basically a combination of about 6 or 7 PCA components on day 4, and so on and so forth. So you can really see that the first PCA component on day 7 is the same, and it's basically always the same. But the other ones are starting to get more spread out, and furthermore, it seems like day 2 and 4 have a more diagonal connection, whereas day 4 and 7 seem to be more spread out, and day 2 and 7 is even more spread out. So on the other end here, if you look at, well, how much of the PCA component can be explained by the first 64 components on another day. If you use day 2 data, you can explain day 4 PCAs according to this blue line, right? And so you can see that actually you can explain day 7 PCAs using day 4 PCA data pretty well, right? So if you use the PCAs from day 4, you're doing a pretty good job. So that shows that the PCAs have not actually changed that much. So it's not really that the PCAs have changed, it's that they're leading to different results, right? So somehow the PCAs, the similar PCAs maybe, have now lead to wildly different results that cannot be predicted from another day, but they do make sense, meaning that they can be predicted from the same day. So the question is, have the cells differentiated, right? Maybe the cells have differentiated, and then it will be a new type of cell that has its own new rules, and maybe those new rules are internally consistent, can be predicted from other cells that have also differentiated, but cannot be predicted from the original stem cells. And so it's a question. It's not. There's no conclusive evidence. We don't have the data to study differentiation. But it's an interesting question. So, yeah. So here, one of the things that we can do is we can just use day 4 data, because day 4 is closer to day 7 than days 2 and 3, and then that actually does a better job than using day 2, 3, and 4 together, just using day 4. All right.